All right, let's jump straight into this high-yield explainer on gametogenesis. Welcome back, Focus Learners. Today, we're diving into a masterclass completely derived from our biological source texts and tailored strictly for your exam preparation. We're going to break down the exact biological processes that create human life, and we're doing it step by step, really locking in on the terminology you absolutely need to know. But real quick, before we get into the weeds, I want to start with a crucial question. At which exact stage is a human egg arrested before puberty? This is a massive trap in medical exams, and it's a detail that trips up thousands of students literally every single year. Well, by the end of this masterclass, not only will you know exactly how to answer this, but you're going to understand the biological why behind it too. To get us there, here's our game plan. First, we'll cover the basics of gametogenesis. Second, the step-by-step male pathway. Third, the female pathway. Fourth, we'll spot the differences between the two. And finally, we'll wrap up with a rapid revision session to really test your active recall. Okay, starting off with the basics. We've got to lay down some biological ground rules and get our vocabulary perfectly aligned before looking at any complex pathways. So what exactly is gametosis? Well, looking at our strictly sourced definition, it's a biological process by which precursor cells undergo cell division and differentiation to form mature haploid gametes. In really simple terms, it's just the ultimate manufacturing process of reproductive cells. We call these gametes, so the sperm in males, and the ovum or egg in females. And that brings us to the absolute most important mathematical rule of this entire topic, ploidy. A diploid cell, which we represent as 2N, contains a full set of 46 chromosomes. A haploid cell, represented as N, contains exactly half, just 23 chromosomes. Vocally tracking whether a cell is diploid, 2N, or haploid, N, at every single step is your core strategy here. If you can track the N, you're going to ace this topic. Moving right along to the male pathway, spermatogenesis. Our source material confirms this process begins specifically at puberty and takes place deep within the seminiferous tubules of the testes. So the whole thing kicks off with the multiplication phase. We start with precursor cells called spermatogonia. Notice they're ploidy, they're diploid, 2N. These cells undergo mitotic divisions just to increase their sheer numbers. Some of them then grow and become primary spermatocytes. Now, listen closely here because examiners love to trick you on this step. The primary spermatocyte is still a diploid 2N cell. The chromosomes haven't halved yet. Once we have that primary spermatocyte, boom, the magic of reduction division begins. Meiosis I kicks in, having those chromosomes to produce 2 haploid secondary spermatocytes. We've officially shifted from 2N down to N. Then these secondary spermatocytes immediately undergo meiosis II, which is an equal division, giving us 4 haploid spermatids. But wait, spermatids are immature. They are definitely not the final mature sperm yet. Getting them to that final stage requires a highly specific process called spermiogenesis. Please do not confuse this with spermatogenesis. Spermiogenesis is the specific developmental phase where those round, immature spermatids morph into highly specialized, functional spermatozoa, complete with a head, midpiece, and tail. Once mature, they're released from the Sertoli cells in a process called spermiation. So doing the math, 1 primary spermatocyte ultimately yields 4 functional sperm. All right, let's switch gears to oogenesis, the female pathway. And what's incredibly fascinating about this next section is how it operates on a completely different, almost interrupted timeline compared to males. While spermatogenesis waits all the way until puberty, oogenesis actually begins during fetal embryonic development. Millions of diploid oogonia form in the fetal ovaries. These cells start dividing and enter into the primary oocyte stage, which is also diploid, 2N. But then, everything just hits the brakes. The primary oocytes get arrested in prophase 1 of meiosis. They literally sit in this state of suspended animation, enclosed inside primary follicles, until the female reaches puberty. Fast forward to puberty, hormonal cycles kick in, and that primary follicle matures into a Graafian follicle. That arrested primary oocyte finally gets to complete meiosis I. But look at this unequal division. It produces 1 massive haploid secondary oocyte and a tiny first polar body that basically just junks the extra chromosomes. Then the secondary oocyte starts meiosis II, but hits a second major roadblock. It gets arrested at metaphase II. This arrest is absolute. The only way meiosis II ever completes is if fertilization happens. Specifically, a sperm cell has to enter. Only then does it divide one last time to form the mature ovum and a second polar body. Okay, now that we've seen both, let's spot the differences. Understanding how these parallel pathways diverge is absolutely crucial for your exams. Let's compare them side by side. First, the anatomical sites. Spermatogenesis happens in the testes, oogenesis in the ovaries. Second, the start times are starkly different. Males begin at puberty, while females start way before birth during fetal life. Third, cell division. Spermatogenesis features perfectly equal cell division, but oogenesis relies on strictly unequal division to produce polar bodies, preserving all that vital cytoplasm for the egg. Next, just look at the massive difference in output. One primary spermatocyte gives you 4 functional sperm, but one primary oocyte? That gives you just 1 functional ovum. Plus, spermatogenesis is a continuous process, churning out millions of sperm daily, driven by testosterone from Leydig cells. Oosiogenesis, on the other hand, produces just 1 ovum per menstrual cycle and eventually stops completely at menopause, which perfectly sets us up for a quick test. All right, time for our rapid revision. Let's lock this knowledge in with a quick recall session, mimicking previous year exam questions. Try to answer these in your head before I reveal the text. Here's your first challenge, and it links all the way back to the very start of our explainer. At which exact meiotic phase is the secondary oocyte arrested, just waiting for fertilization? Did you get it? It's metaphase 2. Remember, the primary oocyte is the one arrested in prophase 1 during childhood, but the secondary oocyte, the one that actually gets released, is stuck in metaphase 2, and that arrest is only broken by the physical entry of a sperm cell. Awesome, let's hit our second high-yield question. How many functional gametes are produced from one primary oocyte? Think back to that unequal cellular division we talked about. The answer is exactly 1 functional ovum. That unequal division is incredibly deliberate. It preserves all the precious cytoplasm and nutrients for that single vital ovum, pushing the excess genetic material into non-functional polar bodies. To make absolutely sure this is cemented in your brain, let's trace the final linear flow one more time. For the male pathway, track the ploidy shift with me. Diploid 2N spermatogonia grow into diploid 2N primary spermatocytes. Meiosis I hits, dropping us to haploid N secondary spermatocytes. Meiosis II gives us haploid N spermatids, which then finally mature into haploid N sperm. And we do the exact same quick recap for the female pathway. Diploid 2N oogonia grow into diploid 2N primary oocytes. After puberty and meiosis I, we get the haploid N secondary oocyte. And finally, if fertilization happens, we get the mature haploid N ovum. I want to leave you with this one-line takeaway that summarizes our entire masterclass. Gametogenesis is the ultimate biological foundation, transforming diploid precursor cells into the mature haploid keys of reproduction. Truly, if you master these fundamental shifts from 2N down to N, you master the core of reproductive biology. But, this brings up a really fascinating, provocative question. What structural changes actually occur in the female reproductive system during the menstrual cycle to prepare for this newly formed ovum? We know the egg is ready, but how does the body prepare a home for it? Well, that is exactly what our next explainer is going to cover. Until then, make sure to like, share, and subscribe to BIOCORE Notes so you never miss a step in your exam prep.